Photosynthesis. Introduction. Types of photosynthesis. Site of photosynthesis. Ultrastructure of chloroplast. Photosynthetic pigments. Photochemical phase light reactions. Pigment systems. Photosystems. Photolysis of water. Hill reaction. Photophosphorylation. Biosynthetic phase. Dark reactions. Significance of photosynthesis. Photosynthesis. The use of light energy to produce carbohydrates from simple inorganic materials like carbon dioxide and water. 6 CO2 plus 12 H2O in presence of light and chlorophyll gives C6H12O6, that is glucose, plus 6 H2O plus 6 O2 gas. Photo light. Synthesis, putting together. Light energy is converted into chemical energy. During this process, the water is oxidized to oxygen, whereas carbon dioxide is chemically reduced to carbohydrate. Oxygen is liberated out in the atmosphere. It is intracellular and anabolic process, constructive process, and ergonic energy storing process. Living organisms can use only biologically usable form of energy, that is ATP, adenosine triphosphate. Types of photosynthesis. On the basis of evolution of oxygen. Oxygenic photosynthesis and anoxygenic photosynthesis. Oxygenic photosynthesis. Oxygen is evolved as a byproduct. The water H2O acts as a hydrogen donor. For example, all green plants and cyanobacteria. Anoxygenic photosynthesis. Oxygen is not evolved as a byproduct. Instead of water, sulfur compounds H2S or non-sulfur compounds act as hydrogen donor. For example, purple sulfur bacteria, chromatium, purple non-sulfur bacteria, rhodospirillum, green sulfur bacteria, chlorobium. Site of photosynthesis. Photosynthesis takes place in green parts of plants. Leaves are the specialized organs for photosynthesis. Green parts of flower like calyx, young green stem, guard cells of stomata also are involved in photosynthesis. These green parts contain green plastids called as chloroplasts. Chloroplasts is the main site of photosynthesis in all green plants, that is algae, bryophyta, pteridophyta, spermatophyta. Chloroplast. Chloros green, plastos molded. Shape. They are polymorphic, that is, are of different shapes like discoid cup-shaped, reticulate, girdle-shaped, stellate, ribbon-like. In higher plants, they are generally spherical or oval-shaped. Distribution. They are mainly present in the mesophyll cells of leaves and young stems of green plants. These are located at the periphery of mesophyll cells with the broad surfaces parallel to the cell wall. This helps in easy diffusion of carbon dioxide, which is required for photosynthesis. Chloroplasts also present in cytoplasm of only the green cells of plants. The distribution of chloroplasts is uniform. Size. Chloroplasts are microscopic. Their size also varies, but the size of chloroplast is constant for a given cell type. They measure about 4 to 10 micrometers in length and 2 to 4 micrometers in breadth. Thickness. Number. The number of chloroplasts per cell varies. In green cells of higher plants, number of chloroplasts varies from 20 to 100. Ultrastructure of chloroplast. Chloroplast is a living cell organelle containing complete machinery for photosynthesis. Chloroplast is externally surrounded by a double membrane envelope made up of lipoproteins, enclosing homogeneous transparent watery substance called stroma or matrix. Within this matrix, grana are present, singular granum. Grana are closely packed stacks 50 to 60 of disc-shaped thylakoids. There are about 40 to 100 grana in chloroplast, and each granum contains about 10 to 100 thylakoids. These grana are connected by intergranal lamellae or stromal lamellae. Thylakoids contain many flattened spheres called quantosomes. These give a granular appearance to the thylakoid. Quantosomes were first noted by Park and Beggins in 1964. Quantosomes contain pigments, electron carriers, coenzymes. Detailed study of each component in chloroplast. Double membrane envelope, stroma or matrix, grana, thylakoids, quantosomes, intergrana lamellae, stroma lamellae. Double membrane envelope, peristomium. Double membrane envelope, peristomium. Outermost protective covering, made up of lipoproteins, lipids and proteins. Double membrane means inner membrane and outer membrane. Both membranes are thin, smooth and continuous. Outer membrane permeable to many solutes and metabolites. Inner membrane selectively permeable. Two membranes are separated by periplastidial space. This space contains periplastidial fluid. Matrix, stroma. Internal space of the chloroplast is filled with a colorless hydrophilic matrix called stroma. It is a protein colloid complex. It contains number of enzymes, granules, DNA, RNA, ribosome. It also contains all the enzymes required for CO2 fixation, which is called dark reaction or biochemical reaction. Rubisco enzyme is mainly present. Enzymes are related to the synthesis of photosynthetic pigments. Calvin cycle, lipid metabolism, synthesis of starch, replication of DNA, transcription, translation. Starch grains are also present, which are the final product of photosynthesis. Chloroplast DNA or ctDNA is usually circular, naked and double-stranded. The DNA of chloroplast is known as plastidome. As the stroma contains DNA and RNA, due to which the chloroplast becomes self-regulating, self-duplicating, autonomous living cell organelle. It is the main site of dark reaction of photosynthesis. Intergrana lamellae, stroma lamellae. Grana are connected with each other with stroma lamellae. Stroma lamellae are colorless membranous structure, also called as fret lamellae. Stroma lamellae contains only PS1 and helps in transport of materials. They contain carbon dioxide fixing enzymes and helps in fixation of atmospheric carbon dioxide. Grana. Grana are suspended in stroma, interconnected by stroma lamellae, intergranal lamellae. Grana lamellae contains both the pigment systems PS1 and PS2. Thylakoids. Thylakos pouch or sac, oid-like, are the structural and functional elements of chloroplast. They are embedded throughout in the matrix. Each thylakoid is a flattened sac lined by a membrane specialized in harvesting light energy. Thylakoids are placed one above the other like a stack of coins. Membrane of thylakoids are made up of proteins and lipid lipoprotein. Layer of chlorophylls and carotenoid molecule is present in between proteins and lipids. A hydrophobic porphyrin ends extends into the aqueous protein layer and lipophilic tails into lipid layers. Carotenoids are sandwiched between the phytol tails and phospholipids molecules. Quantosomes, photosynthetic units. These are present in thylakoid. They are small, rounded, flat structure. They measure about 180 into 110 into 120 Armstrongs. A group of pigment molecules necessary to effect a photochemical act, that is, the release of one molecule of O2, is called a photosynthetic unit. These particles are responsible for absorption of quantum of energy, so they have been called quantosomes. Quantosomes contain about 230 to 300 chlorophyll molecules. They are arranged as a single layer on the thylakoid bodies. Photosynthetic pigments. Photosynthetic pigments are the pigments involved in the process of photosynthesis. They are organic molecules which absorb light of specific wavelengths in the visible region. Pigments are present in the quantosomes of thylakoids in chloroplast. There are two major groups of photosynthetic pigments. Essential pigment, chlorophyll A. Accessory pigments, chlorophyll B, carotenoids and phycobilins. Chlorophylls. Chlorophylls, Greek chloros green, phyll leaf, are the green pigments present in the photosynthetic autotrophs except bacteria. The green pigments were named as chlorophylls by Pelletier and Caventry in 1817. C.G. Stokes, 1864, separation of pure fractions of chlorophyll A and chlorophyll B. Chlorophylls absorb light in the violet, blue and red wavelengths of visible spectrum of light and reflect green light. This is the reason they appear green. Chlorophylls are chelate salts of magnesium and insoluble in water. They are soluble in organic solvents like ethyl alcohol, acetone, petroleum ether. There are 7 types of chlorophyll. Chlorophyll A is present in photosynthetic plants. B in green algae, bryophytes, pteridophytes and spermatophytes. Chlorophyll C is present in dinoflagellates, diatoms and brown algae. Chlorophyll D is present in red algae. Chlorophyll E is present in yellow-brown algae. Bacteriochlorophyll and bacterioviridin or chlorobium. Willstater, Stoll and Fischer in 1912 studied the structure of chlorophyll. It is an asymmetrical molecule having head and tail. There are green pigments with polycyclic planar structures. The head is a tetrapyrrole ring or 4 porphyrin rings with non-ionic Mg atom in the center. The 4 pyrrole rings of head are connected with each other by methane groups, forming a ring structure. Head is also called as a porphyrin head. It is polar and hydrophilic. Each pyrrole ring is made up of 4 carbons and 1 nitrogen. Nitrogen of all the 4 pyrrole rings are towards the center of porphyrin. Non-ionic Mg atom in the center is attached to the nitrogens of all the 4 pyrrole rings. Tail is a single, long phytol chain attached to the 4th pyrrole ring. Tail is non-polar, hydrophobic and extends into lipid layer of the thylakoid membrane. Main difference between structure of chlorophyll A and B is chlorophyll A has a methyl group. Chlorophyll B has an aldehyde group at carbon 3 of pyrrole ring 2. A chart showing various pigments, pigment color, molecular formula and their solubility and insolubility is given below. Pigments. Chlorophyll A. Role in photosynthesis. It is an essential pigment. It catalyzes. Phosphorylation. Acts as an antenna molecules in trapping light energy. Reaction center for PS1. Reaction center for PS2. Chlorophyll B. Role in photosynthesis. Accessory pigment. Collects light energy and transfers it to chlorophyll A. Carotenoids. Carotenes and xanthophylls. Phycobilins, biliproteins, phycocyanin, phycoerythrin. Role in photosynthesis. Accessory pigment. Collects light energy and transfers it to chlorophyll A. Protects chlorophyll from photooxidation. Carotenoids. These are lipid compounds widely distributed in plants, animals, microorganisms such as photosynthetic bacteria and fungi. All the carotenoid pigments have 40 carbon atoms. These are accessory pigments and absorb light energy in the mid-region, blue and blue-green regions of visible spectrum and transfer their absorbed energy to chlorophyll molecules. They absorb the wavelengths which are not absorbed by the chlorophylls. Carotenoids are soluble in organic solvents like acetone, chloroform and lipids, whereas insoluble in water. They pick up nascent oxygen released during photooxidation of water and change them into harmless molecular state. Thus, they protect chlorophyll molecules from photooxidation. Carotenoids are also present in chromoplasts in flowers and fruits and make them conspicuous to animals for pollination and dispersal. A chart giving the characteristics of carotenes and xanthophylls is given below. Phycobilins, biliproteins. These pigments absorb light and transfer it to chlorophyll A and therefore considered as accessory pigments. These are found only in blue-green algae, cyanobacteria and red algae. These are water-soluble pigments and are thought to be localized in small granules called phycobilisomes attached to the thylakoids. These are stable and crystalline chromoproteins. They do not bear magnesium atom and phytol tail. A chart showing characteristics of phycocyanin and phycoerythrin is given below. Role of photosynthetic pigments. All pigments absorb solar energy of different wavelengths. Chlorophyll A and chlorophyll B absorb red light having wavelength 600 to 700 nanometer. Carotenoids, carotene and xanthophyll absorb violet to blue light having wavelength 400 to 420 nanometer, and phycobilins, phycocyanine absorb orange light 615 nanometer. Phycoerythrin absorbs yellow light 560 nanometers. Accessory pigments chlorophyll B, carotenoids and phycobilins act as antennae to absorb and transfer solar energy to chlorophyll A. Chlorophyll A absorbs energy as well as functions as a reaction center to convert radiant energy into chemical energy. Chlorophyll A ultimately receives all energy from all the pigments and gets excited, releasing high energy electron, which is utilized in the formation of ATP. Chlorophyll A brings about splitting of water into hydrogen and oxygen. Destruction of chlorophylls by light in presence of oxygen is called photooxidation. Carotene and xanthophyll act as guard pigments by preventing photooxidation of chlorophyll A. Nature of light. Sunlight in the visible spectrum of electromagnetic radiation is the main source of energy of photosynthesis. The spectrum of visible light ranges from wavelength 400 nanometers violet to 700 nanometers red. A small part of electromagnetic spectrum consists of waves of 7 colors: violet, indigo, blue, green, yellow, orange and red. Photosynthetic pigments absorb light spectrum between 400 and 700 nanometers is called as photosynthetically active radiation, PAR. The wavelength of light is ultimately made up of tiny particles called photons. The unit quantity of light energy is called quantum. Photon is equal to 1 quantum of energy. The amount of energy in each quantum of light is inversely proportional to 1 upon wavelength of light. Light is a continuous beam of discrete photons. The light with shorter wavelength has greater energy and the light with longer wavelength has lesser energy. Blue light, wavelength 390 nanometers, has greater energy, 70 kilocal. Red light, wavelength 700 nanometers, has lesser energy, 40 kilocal. Red and blue light are the most effective for photosynthesis. Green light is less effective. It is not absorbed but is emitted out. Why do chlorophyll molecules use light in visible wavelength for photosynthesis? Chlorophyll molecules absorb light only in the form of photons. The energy in a mole of photons, 1 Einstein, that is 6 into 1023 photons, is 170 to 300 kilojoules, about an order of magnitude more than the energy required to synthesize a mole of ATP from ADP and PI, about 30 kilojoules under standard conditions. The energy of 1 Einstein in the infrared or microwave regions of the spectrum is too small to be useful in photosynthesis. UV light and X-rays, on the other hand, have so much energy that can damage proteins and nucleic acids and induce mutations and are often lethal. Therefore, light in visible wavelength are used for photosynthesis. Synthesis of chlorophyll. In flowering plants, chlorophyll synthesis takes place in the presence of light. Synthesis can also occur in absence of light in the plants like algae, bryophytes, pteridophytes and gymnosperms. Mechanism of photosynthesis. The process of photosynthesis is a redox process and is completed in 2 phases. Phase 1, light reaction or photochemical phase. Phase 2, dark reaction or biochemical phase or thermochemical phase or synthetic phase. Photochemical phase, light reactions, also known as Hill reaction. Photochemical is the first phase of photosynthesis. It is dependent on the light, hence called light reaction or photochemical phase. Light phase involves absorption of light energy by photosynthetic pigments and its conversion into chemical energy or ATP and photolysis of water, leading to formation of NADPH2 and liberation of oxygen. In this phase, the assimilatory power in the form of ATP and NADPH2 is generated in the thylakoids. Photochemical phase mainly involves formation of ATP, photophosphorylation, photolysis, oxidation of water, liberation of oxygen and formation of NADPH2 reduced coenzyme. Formation of ATP, photophosphorylation. Process of formation of ATP from ADP and inorganic phosphate in presence of light and chlorophyll discovered by Arnon. In other words, it is a process of conversion of light energy into chemical energy. Photophosphorylation is of two types, cyclic and non-cyclic. Photolysis, oxidation of water or Hill reaction. Initial assumption: oxygen was evolved by splitting of carbon dioxide and not splitting of water. Van Niel in 1931, while experimenting with green sulfur bacteria, postulated that during photosynthesis, water splits and not carbon dioxide. Robert Hill experimentally proved Van Niel's postulation. Martin, Rubin and Kamen confirmed Robert Hill's findings. Robert Hill experiment 1937. Suspended chloroplast in water with electron acceptor ferricyanide and oxygen acceptor hemoglobin. The sample was illuminated in absence of carbon dioxide. At the end of the reaction, ferricyanide was reduced to ferrocyanide and hemoglobin was converted into oxyhemoglobin. This proved that it is not carbon dioxide but water that splits during photosynthesis to form oxygen. Thus, he proved that water splits and the source of oxygen is water, not carbon dioxide. This splitting of water in the chloroplast is called as photolysis of water or Hill reaction. 2H2O plus 2A in presence of light and chlorophyll gives 2AH2 plus 6O2. A is the unknown hydrogen acceptor present in the chloroplast of plant cells. Rubin and Kamen in 1941 confirmed and directly proved that source of oxygen evolved during photosynthesis is water. Dr. Anand and his co-workers in 1954 discovered the unknown substance A in Hill's reaction, working as hydrogen acceptor in plant cells. It is a coenzyme called nicotinamide adenine dinucleotide phosphate, NADP. Hill's reaction can be rewritten as 2H2O plus 2NADP in presence of light and chlorophyll A gives 2NADPH2 plus O2. Pigment systems, photosystems. It is a group of photosynthetic pigments which absorb light energy and convert into chemical energy. Two pigment systems are involved in the light phase of photosynthesis, proved experimentally by Emerson. These photosynthetic pigments are present in quantosomes. In each photosystem, there is a single molecule of chlorophyll A which receives the light energy from accessory pigments and releases electron. This molecule is called reaction center. Depending on the reaction center, there are two types of photosystems, photosystem 1 and photosystem 2. In photosystem 1, PS1, its reaction center is chlorophyll A 700 or P700. P pigment and 700 maximum absorption at 700 nanometer wavelength of light. PS1 is located on the outer surface, activated by the light of wavelength longer as well as shorter than 680 mu. In photosystem 2, PS2, its reaction center is chlorophyll A680 or P680. P is the pigment and 680 the maximum absorption of 700 nanometers wavelength of light. PS1 located on the inner surface of the thylakoid membrane, activated by the light of wavelength shorter than 600 mu. Photosystem along with proteins form a pigment-protein complex PPC in the thylakoid membrane, which has a co-complex CC, one chlorophyll A molecule and other chlorophyll molecules, electron acceptors and donors, and light harvesting complex or antenna complex, which has the rest of chlorophyll and accessory pigment molecules. A chart showing difference between photosystem 1 and photosystem 2 is shown below. Non-cyclic photophosphorylation. This process involves both the photosystems, that is photosystem 1 and photosystem 2. Reaction center of PS1 is P700 and reaction center of PS2 is P680. Both photosystems have chlorophyll A and accessory pigment molecules. The reaction center of PS2 P680, chlorophyll A680, gets photoexcited, activated on receiving photons of light. It expels energy-rich electrons and becomes ionized, P680+. The electron is immediately accepted by the primary acceptor, plastoquinone. The reaction center P60+ now has become oxidized and can't participate in light reaction again until it comes to neutral state, and this can be achieved by gaining electrons. From where will reaction center get electrons? Answer to this question is water. Photolysis of water. That is, splitting of water takes place as shown in the diagram, and due to this splitting, electrons are released and are accepted by P680+, and it becomes neutral, that is P680, and can once again participate in a light reaction. During this splitting of water, O2 is also evolved. Meanwhile, the electron expelled from P680 and accepted by plastoquinone and then to cytochrome B6, cytochrome F, plastocyanine. During this journey, at each step of electron transfer, the electrons lose potential energy. This energy is utilized in the formation of ATP from ADP and PI, that is inorganic phosphate. This occurs when the electron ID transferred from cytochrome B6 to cytochrome F. From plastocyanine, electrons of PS2 pass to PS1, that is P700. When PS1 is illuminated by light, P700 is excited and emits out energy-rich electrons, which is first accepted by ferredoxin FD via ferredoxin-reducing substance FRS, then to ferredoxin and then NADP. When NADP gets electrons, it gets ionized. Thus, the electron does not go back to P680. Thus, this is a non-cyclic process. The hydrogen ions, which are released during photolysis of water, go to ionized NADP to form NADPH2. Significance: it produces ATP molecules which act as energy donors for dark reaction. It also produces NADPH2, which acts as hydrogen donor for dark reaction. It releases O2 as a byproduct by the process of photolysis of water. Cyclic photophosphorylation. In this pathway, the electrons expelled from P700 return to P700. The light energy is absorbed by pigments in light harvesting complex associated with PS1. Energy is transferred to reaction center P700. It gets excited and energy-rich electrons are expelled from P700, due to which it becomes positively charged. Electrons are accepted by an ion-containing red protein called ferredoxin FD via ferredoxin-reducing substance FRS. These electrons pass through a series of electron acceptors and donors, carriers such as cytochrome B6, cytochrome F and plastocyanine PC. During this journey, at each step of cyclic electron transfer, the electrons lose potential energy. This energy is utilized in the formation of ATP from ADP and PI, inorganic phosphate. This occurs when an electron ID transferred from cytochrome B6 to cytochrome F. From plastocyanine, the de-energized electrons are transferred to P700. As P700 gets back the expelled electrons, the process is said to be cyclic. Significance: light energy is converted into chemical energy in the form of ATP. Does not involve PS2, hence no photolysis of water is there. Hence, no release of oxygen or NADPH2 is formed. It takes place when non-cyclic process stops due to certain reasons. A chart showing comparison between cyclic photophosphorylation and non-cyclic photophosphorylation is shown below. Dark reactions, biosynthetic phase, secondary processes, synthetic phase. Light is not essential. Takes place in stroma of chloroplast. Brings about reduction of carbon dioxide to carbohydrates. First established by Blackman in 1905. Hence, it is also called as Blackman reaction. Dark reaction utilizes end products of light reaction, ATP and NADPH2, to reduce CO2 into carbohydrates. Each step of reaction is controlled by specific enzyme. Path of carbon in the dark reaction was traced by Melvin, Kelvin and Benson. The pathway is called as Kelvin cycle or C3 pathway. After fixation of CO2, the first stable compound formed is 3,C-phosphoglyceric acid, PGA. Hence, it is also called as C3 pathway. Kelvin cycle is also known as C3 Kelvin-Benson cycle, synthetic phase, photosynthetic carbon reduction cycle. Calvin cycle operates in 4 steps: carboxylation, that is fixation of CO2, reduction of PGA, that is glycolytic reversal, formation of hexose sugar, that is the synthetic phase, and regeneration of RUBP. Calvin cycle. Carbon fixation. 3 molecules of ribulose monophosphate RUMP 5C compound are phosphorylated by ATP to form 3 molecules of ribulose diphosphate RUDP. RUDP is the first carbon dioxide acceptor. CO2 comes into the stroma of the chloroplast via the stomata of the leaves. It absorbs CO2 in presence of RUBP carboxylase or Rubisco and forms additive 6-carbon compound which is unstable, which immediately splits in half to form 6 molecules of 3-phosphoglyceric acid. Reduction. 6 molecules of 3-phosphoglyceric acid are further phosphorylated by ATP and converted into 6 molecules of 1,3-diphosphoglyceric acid DPGA. 6 molecules of 1,3-diphosphoglyceric acid are further phosphorylated by ATP and converted to 6 molecules of 3-phosphoglyceraldehyde PGAL by utilizing H2, NADPH2 and releasing 1 phosphate each. 1 molecule of 3-PGAL, a triose sugar, can be directly used for metabolism of the plant, or it can be converted into hexose sugar and is ultimately stored in the form of starch. Regeneration. 5 molecules of 3-PGAL, 3C, are converted into 3 molecules of RUMP, 5C, through intermediate products like ethyrose and pseudoheptulose to continue the cycle. Significance of Calvin cycle. By using ATP and NADPH2 formed in the light reaction, CO2 is reduced to form organic food in the form of 3-phosphoglyceraldehyde 3C or glucose 6C, which can be utilized directly in plant metabolism or can be stored in the form of starch. ATP and NADPH2 are utilized as energy donor and H2 donor to form ADP and NADP respectively for reuse in the light reaction. A chart showing the differences between the light reaction and the dark reaction is shown below. Hatch-Slack and Kortschak HSK pathway or C4 pathway. In certain tropical plants, CO2 is not directly absorbed by Rubisco. In these plants, due to high temperature and high light intensity, keep their stomata partially closed, due to which the CO2 concentration goes down and Calvin cycle cannot be started. Therefore, they follow another pathway. In this alternative pathway, the first stable compound, it is 4C compound, oxaloacetic acid, and hence it is called a C4 pathway. Plants like maize, sugarcane, jowar, euphorbia, amaranthus, portulaca, etc. have this C4 pathway. Hence they are called as C4 plants. H.P. Kortschak, 1965, reported this alternative method of CO2 fixation for the first time in sugarcane. In 1970, M.D. Hatch and C.R. Slack outlined the entire series of reactions. Hence it is called Hatch and Slack pathway or HSK pathway. The plants following C4 pathway show Kranz anatomy. Kranz meaning wreath or necklace. That is, the vascular bundles are surrounded by bundle sheath cells forming a wreath-like layer. Around the bundle sheath cells are the loosely arranged mesophyll cells. Chloroplasts of bundle sheath cells are agranal, that is, without grana, and chloroplasts of mesophyll cells are granal, that is, with well-developed grana. Thus, chloroplasts show dimorphism. C4 plants follow C4 pathway in which CO2 is absorbed by PEP, phosphoenol pyruvic acid, in presence of enzyme PEP carboxylase. This enzyme, being sensitive, can absorb O2 of as low concentration. CO2 of low concentration enters the mesophyll cells through stomata and is absorbed by phosphoenol pyruvic acid 3C with the help of enzyme PEP carboxylase to form oxaloacetic acid 4C. Oxaloacetic acid 4C, being unstable, gets converted into stable acids like malic acid or aspartic acid 4C. These acids diffuse into bundle sheath cells where they release CO2 to form pyruvic acid 3C. When enough CO2 is accumulated, it is absorbed by RUDP carboxylase of Calvin cycle, while pyruvic acid diffuses back to mesophyll cells. Pyruvic acid 3C is phosphorylated by ATP to form phosphoenol pyruvic acid to continue the HSK pathway. The significance of photosynthesis is given below.